we'll take a brief moment here with a part 10 to summarize the major events of mitosis. So here's a cell that is about to enter into mitosis, what I like to call dosage regulation. So we have enough here to create 2 cells, and we're going to get the centrioles, which have also replicated, dividing. They're going to move out toward their poles. We're going to have the breakdown of the nucleus. It's going to dissolve, literally. Okay, we have the condensation of the mitochondria. And then we have a formation now of these spindles which reach out to the replicated mitochondria. I'm sorry, to the replicated DNA molecules. And there's a little chunk of protein here called a kinetochore that has held the two, how shall I say, the fully replicated chromosomes together, right? So we've just sort of bundled them right here in the middle with a little piece of protein, and the name of that protein is called kinetochore. Okay, and actually the microtubules attach to the kinetochore. That's how they're managing the position. So now we have everybody in position right here, and this is an important decision-making process right here. The metaphase is where we are along the equatorial plane. So there's going to be a cell over here, and there's going to be another cell over here, and we get down to dividing the spoils, as it were. Right, so this is the ultimate estate moment between the the inheritors. So what's going to happen is that we're going to break the kinetochore, and we're going to pull half of the information, the replicated DNA, to one side, and then the other half of the DNA to the other side. Okay, so this is a late phase here, or anaphase. It's late metaphase, or you could call it anaphase, and it's sort of like grabbing your child, to pull them across the street before the light turns, and you can see the little flailing of the arms right there. And so you're pulling right here. So the kinetochore has actually divided, and the spindle is pulling the molecule of DNA, which represents the chromosome, to the opposite side. Finally, we have completed our dosage regulation, and the membranes are now reforming. There's a membrane, cell membrane, which then divides this sort of Caesarean twin right here into 2 completely separate cells. And that completes our project. Okay, so I'm not going to go back to the transcription translation thing. At some point you'll be taking a microbiology course, and you'll revisit this transcription translation episode right here, which is really quite a lot of fun. Okay, so that completes our project for this chapter, number 3, and we're moving on next to chapter number 4, which is going to be going from cells then to tissues. Tissues make up the cells, and so we'll be preparing you to introduce to the chapter number 4 right here. Okay.